Hello, everybody, and welcome to today's webinar. I see we have participants from all over the world. I see some familiar names, lots of new friends. Very excited for today's webinar. Today, we'll be highlighting a new addition to the Twist library product family, spread out low diversity libraries. I'm Stephen Thomas, Business Development Manager for Twist Biopharma. Before we begin today, I would like to cover a few housekeeping items. All lines will be muted during the webinar. We will answer all questions after the presentation. If you have a question, please submit in the Q&A box rather than the chat box. At the completion of the webinar, you will be redirected to take a brief survey. We would greatly appreciate your feedback as it will help us improve the quality of future webinars. Today, we have the great pleasure of hearing from Library's product manager, Lucy Zhu. Lucy graduated from UC San Diego with a BS in Bioengineering Biotechnology. Lucy joined the Twist Biosciences research and development team in 2016 when it was first developing combinatorial variant libraries. She's collaborated with leaders from small biotechs to big pharma companies to build custom libraries that precisely target their needs. Since then, she's led development efforts to further grow the site variant and combinatorial variant library products to tackle challenges in antibody engineering, protein engineering, and directed evolution. With that, Lucy, you have the floor.
Good morning, everyone. Thank you for joining me today. I'm very excited to share with you what we've been developing here at Twist. I'm going to give you an overview of how we've leveraged our technology to build DNA variant libraries in a quick and inexpensive manner so you can have the tools you need in your hands in about 2 weeks. The product that I'll be talking about today is our spread out low diversity libraries, or SOLT libraries. We've developed this product for our customers who are doing enzyme evolution and needed a way to rapidly and precisely investigate and optimize the protein of interest. So it looks like a lot of you here today are working on protein engineering, which is perfect. So I'm very excited to share with you how SOLT can play a role and help you do your research a little bit faster and more precise. All right. So I would like to start off by talking to you about our Twist technology. Our ability to write DNA on silicon is the foundation of our story at Twist. This is our core technology that we've used to build all of our other products, and it's what sets us apart. So traditionally, you would need an entire 96-well plate of oligos to build just one gene. Our silicon platform enables us to miniaturize that process and print up to 1 million oligos on a silicon chip. We can print discrete oligos on each of these clusters, which is what you see here on the right-hand side. We then use these clusters of DNA to construct our clonal genes, which many of you are probably very familiar with. And in addition to that, we have a line of NGS products that leverages this capability for target enrichment. For my team, we leverage this technology to make DNA libraries for applications such as protein engineering, antibody engineering, and enzyme evolution. This is a game-changing technology because it enables us to make libraries that are unprecedented in the industry, and our platform allows every customer to design each variant base by base and precisely incorporate the diversity into their region of interest. We've worked with big and small pharmaceuticals and biotechs to use these libraries to build phage display antibody libraries, libraries for scaffold engineering, libraries to study genetic variants, and for protein engineering. As you can see, there are many possible applications. So there are many definitions for what people call a library, but for the purpose of this presentation, a DNA variant library is a collection of sequence variants or gene mutants related to and built from a common wild-type sequence. We have our site variant libraries here, where only a single codon or amino acid is mutated at a time. You can use this to build an alanine scan library or saturate each position with all 20 amino acids. We have used these libraries to study GPCRs to evolve novel glucose sensors. Next, we have our combinatorial variant library, where there are multiple mutations being introduced to the gene. At its simplest form, this type of library can have a few sites mutated simultaneously, which is what you see here. And this can be used for FC engineering. Now, to go one step further, these mutations can also be along a stretch of the gene, and this is what we call a domain. This is typically used to target specific CDRs in an antibody library. We're able to go even further and synthesize DNA variants with insertions and deletions in a single pool for customers who are interested in building length variation libraries. This is advantageous for studying the CDRs of an antibody, and I'll dive a little bit deeper into this in the next couple of slides. So our spread out low diversity libraries, or SOLD libraries, is an expansion of the multiple site combinatorial variant library. Our customers wanted a library with not just a couple of variant positions mutated, but with many variant positions scattered across the gene because they were doing enzyme evolution. So they were designing oligos and primers to build these libraries themselves, and were spending a significant amount of time doing the oligo design and the library build. So although the build process isn't complex, it was time-consuming to set up and execute. They couldn't be exactly sure which variants were incorporated into the final library, and they needed these libraries fast and ready for cloning. So at Twist, we're very excited to announce the launch of our SOLD libraries, because now we have a product that addresses each and every one of those challenges. We've developed a unique way to build a library up to 10 to the 8 in diversity in just about 2 weeks. And it doesn't matter if you need 1 or 10 libraries, we can build them all in parallel. So what we're doing is leveraging our library design pipeline and coupling it with our in silico DNA synthesis platform to build a library that enables our customers to investigate and optimize the activity of a protein both rapidly and effectively. With all of our libraries, you can precisely define the variants at the DNA level. With site variant libraries, you can introduce change to one position of the sequence at a time. It could involve saturating a position with all 20 amino acids, or introducing anywhere from a single codon change to all 64 codons. Our library platform gives you the flexibility to use your own custom codon tables in the library design. And because of this, you can codon optimize the library for any expression system. You can introduce precise codon mutations to a DNA sequence and expect to see a uniform distribution of the variants. This eliminates the codon bias that you would typically see with other methods. Since each variant is designed in silico, we can pre-screen these variants for restriction sites that you might want to use for cloning or any other downstream process. If a restriction site exists, another codon for that amino acid will be substituted in so that you can keep the diversity that you design. This is a great tool, not just for protein engineering, but also for clinical studies to investigate genetic variants that otherwise might be too low to detect. So combinatorial variant libraries, on the other hand, enables you to introduce multiple mutations to the wild-type sequence simultaneously. These mutations can be localized in regions called domains, which is what we see here, and these are short stretches of DNA ranging from a couple of amino acids in length to up to 30 amino acids long. Here is an example of one of our most common customer order product, a VHH antibody library. The library here consists of 3 variant domains, CDR1, 2, and 3, and these are stretches where the diversity will be incorporated. There are many ways to build this type of library, and the reason why our customers are excited about our VHH libraries is because they're able to precisely incorporate the desired mutation into the library without any unintended mutations, codon bias, or stop codons. Our library technology enables us to incorporate length variation as well into the library, which is particularly exciting for those who are interested in investigating the different length profiles of the CDRH3 loop, or for those that want to replicate the CDR profile observed in the human repertoire. So instead of ordering a library for each of these different lengths and then pulling it together, our customers are able to design and order it all in a single pool, which reduces the cost on their end. But what really sets us apart is our ability to help customers remove liabilities, non-specificity motifs, and any other properties that they don't want in their library. as you can see here. So they can remove motifs that are known to cause developability issues, so the library is enriched for variants that are more likely to succeed. And our ability to offer precise control over the library diversity empowers our customers by giving them the ability to screen exactly what they want and screen more functional variants. So our spread out low diversity sits in the middle between our SVLs and CVLs. This type of library provides a quick and precise tool for exploring the sequence space encoded by mutating multiple positions simultaneously scattered across the wild-type sequence. So unlike the SVLs, where you're just mutating a single position at a time, and unlike the CVLs, where the diversity is often confined to these domains, a sole library, you can incorporate diversity that's scattered along the wild-type sequence. So our team has developed a unique way to build this library that enables us to construct a library in about 2 weeks without any template. And one of the main applications of this library type is enzyme evolution. Enzyme evolution and protein engineering have been widely used strategies for the development of enzymes with improved functions such as higher specificity, thermostability, and performance. It's used in metabolic engineering and industrial biocatalysis to improve product yields and reduce losses to side products, which is what some of our customers are using the SOL libraries for. So with this library, we aim to help enable these workflows in a more rapid and controlled manner. This library is not just limited to enzyme evolution. We also have customers who are using it for framework maturation as well. So we are leveraging our ability to synthesize each variant base by base and precisely incorporating it into the desired diversity in the region of interest with unprecedented uniformity. Before I dive in deeper into our SOL libraries, I wanted to give a quick overview of a few methods that are commonly used to generate these gene libraries. So many of you probably already know random mutagenesis is a commonly used strategy. This is often done in the form of error-prone PCR to generate diversity at random positions. The addition of manganese and biased concentrations of DNTP is one way random mutations can be introduced to the gene sequence. Ideally, this approach will result in randomization along the entire sequence, resulting in mutations that are equally represented. However, this relies on all possible mutations occurring at the same frequency, and some types of errors, and sometimes certain types of errors in the amplification process are more common than others, which leads to bias in the library composition. Then there's also codon bias that results from the nature of the genetic code, where an amino acid can be coded for by more codons than others. So these single nucleotide mutations can lead to bias in the variant amino acids of the sequence as well. Next, we have directed mutagenesis, which is done in a more controlled fashion and targets the specific positions of the sequence. This can be done with degenerate primers, but this often leads to codon bias depending on how these primers are designed. This approach can also be done using our site saturation variant library platform if you're interested in mutating just one position at a time. Our platform enables us to pre-design each variant and synthesize them base by base, which removes unwanted codon bias and stop codons from the library. And finally, we have DNA shuffling, which combines the diversity from different sequences and mixes them in novel combination. So these gene fragments are generated by digesting the source DNA molecule of DNase and then reassembling back together. The difficulty with this method is that it relies on the annealing of the growing DNA strand. So these gene fragments will recombine when they're similar enough to allow for annealing. But regardless of that, it's an effective way of combining sequences with high homology. As you can see, there are many tools that can be used and has been used to generate these gene variant libraries. However, our SOL libraries offer a way to generate these libraries rapidly without the need for a template, and while leveraging our ability to precisely print each variant base by base, pre-screening them for unwanted restriction sites and motifs before assembling them into the full construct. So like all of our library products, SOL libraries enables customers to focus only on the variants with the desired mutations. With random mutagenesis, you don't know what variants are in your library, and due to the bias in the library, some variants that yield beneficial properties may not even be present. Our ability to design each variant in silico enables you to precisely target the desired mutations. This is particularly exciting if you're interested in investigating a variant space that's outside of an existing patent or want to avoid distinct mutations that's covered by it. And unlike traditional methods of library assembly, you can expect to see a uniform distribution of each amino acid at each variant position. So, for example, if a variant position contains 2 amino acids, for example, let's look at position 29, and we want to introduce a histidine and a valine, we would expect to see each amino acid at 50%. And this is exactly what you see with twist libraries. This is made possible because we print each variant base by base, which enables us to control the diversity and eliminate any unwanted bias. So here and today we'll be looking at a case study that we did. We'll be looking at a customer order with a library that has about 26 variant positions. Each position of the library has either 2 or 3 mutations, including the wild type amino acid. And as you can see here, each of these green blocks indicates a real variant position, and most of the diversity is scattered along the sequence, although we do have a bit of it saturated and localized in the center and the end. So we took the customer design and pre-designed all the variants in silico, check that they don't contain any unwanted restriction sites before they're printed. And for variants that do contain a restriction site, our pipeline will substitute in a different codon for the amino acid so that the diversity is preserved and the variant won't be discarded. This is one of the advantages of being able to print the variants base by base. We are not restricted by codon bias or the sets of codons that can be used to build the variant. You have the flexibility to codon optimize the variants before synthesis, and you know exactly what variants have been synthesized and incorporate into your library. So the negative data that comes out of your library can be useful and used for the next iteration of your library design. So here we leverage our low error rate for oligosynthesis to build the library completely synthetically without any template, which enables us to complete the build in under 2 weeks. Here we are looking at just a snapshot of the library, and this diagram isn't to scale, but I hope it gets the point across. We're looking at amino acid distribution in one region where there are 5 variant positions, which is what you see here, and each of these positions contain 2 substitutions. So each color here represents a different amino acid. And since there are 2 substitutions per position, we would expect to see 50% of each amino acid. And when we look at the expected and the observed amino acid distribution, we can see that the observed amino acid distribution matches exactly what the customer had designed. And this shows that at the amino acid level, we are seeing precise incorporation of the desired amino acids. There is no unintended mutations or codon biases as you would see with degenerate codons. There is unprecedented codon control and only incorporation of the desired amino acid. This means that you don't need to do more screens to see the desired variants, and you don't have to screen through non-functional changes. Here, we're going one step deeper and looking at the distribution of variants in that region. So if you remember, we are looking at 5 variant positions, each with 2 substitutions. And this gives us a total of 32 unique variants in that region. So what we did is we took the reads and mapping to that region and analyzed it to look at the uniformity of the incorporated variants. So in a perfect world, the normalized distribution of each variant would be 1. And what this means is that if we have 100 reads and there are 4 variants, we would expect to see 25 reads mapping to each variant. However, everything would be observed at the same frequency. However, this is unavoidable due to limitations in either oligosynthesis, sequence complexity, PCR amplification, or NGS. And we pride ourselves in industry-leading oligo uniformity, which is how we're able to keep the variant distribution very tight. As you can see here, 50% of the variants are observed at the same frequency, and 40% of the other variants are observed at just 0.1x higher or lower than the average. And this indicates that when the library is screened, each variant has a very similar likelihood of being observed. And this shortens the time needed to screen the library. So there are no dropouts in the library, and you know exactly what variants exist in your library because they've been designed in silico and synthesized individually. So by analyzing the amino acid distribution at each position and the variant distribution at each position, we can confirm that we're seeing a uniform incorporation of the requested variants at both the amino acid level and the variant level. There were no unintended mutations and there were no dropouts, so you are screening for variants that you want. Next, let's look at a region that has a higher diversity. So even with regions of higher diversity and sequence complexity, we still see a tight distribution of the desired variants. And we also see no dropouts. So here we're looking at this region of the library where there are a total of 8 variant positions with 2 to 3 mutations each, which gives us a total of 384 unique variants in that region. If we look at the chart here, each dot here represents a unique variant. And again, each variant is synthesized base by base, screened for restriction sites and liabilities before they are incorporated into the full construct. And when we look, so what we did is that we took the recounts that map to this region and calculated the frequency of each of these variants. So despite the sequence complexity of this region, we still see a tight distribution of all of the design variants, and we don't see any dropouts. The lowest observed variant is seen around half as frequently as the average, and all the diversity has been incorporated. So variant uniformity is important because libraries with underrepresented variants will require more time and resources to be spent in addition to screening in order to capture the complete diversity of the library. So we've been looking at small snapshots of the library. Now I want to take a look at the entire construct itself. So here we're looking at the incorporation of variants across the entire construct. So in each of these, we're looking at a distinct snapshot of the construct. And with region 1, you can see that there's 4 variants, and region 4, which is what we just looked at, it contains 384 variants. So as we look across the entire construct, you can see that there is a uniform distribution of all the variants, of all the unique variants in each of these regions. All the variants that are observed at a frequency that is within plus or minus 0.5x of the average. As you can see, when we look across the entire construct, you don't see any dropouts at all. So the more uniform your library is, the less work you will need to do with screening, and this reduces the effort you need to spend to capture the entire diversity of the library. When you have variants that are underrepresented in a library, you'll need to do more screens to capture the diversity, and as you can see, that is not the case here. So here we wanted to look at the construct through a few different windows. So if you remember earlier, we were looking at window 1. Now we want to take that same concept and move that into a sliding window approach. So here, when we look at window 2, we're looking at the variant combinations that would exist if these variants were to be incorporated together. And as you slide this window across, you still see a uniform incorporation of all the diversity that would exist across this space. And as you move it across each of these sliding windows, you still don't see any dropouts. So what this indicates is that we're seeing a complete incorporation of the 10 to the 8 diversity, and we're seeing all the variants, we're seeing all combinations of the variants with each other. And this indicates that the library that the customer has designed is what they received in their hands. Now, our customers came to us with a request for a library product that they could use for enzyme evolution. They were designing oligos and primers to build these libraries themselves, and were spending a significant amount of time doing the oligo design and the library build. And again, although the build process isn't complex, it was time consuming to set up and execute. So we are very excited to now have a product that addresses each and every one of those challenges. Sold libraries leverages the best of Twist technology, which is the ability to precisely define the targets of interest and to get a library that not only that contains only the desired mutations. Customers no longer have to screen through non-functional variants, and they're empowered with a tool that enables them to investigate and optimize the activity of a protein both rapidly and effectively. And although this product was designed with enzyme evolution in mind, our early access customers have also used it for antibody framework optimization. As you can see here, there are many applications for the sold libraries, whether it's enzyme evolution, protein engineering, protein solubility, or antibody framework optimization. We can work with you to identify the best tool for you. So our saying is, you design it and we build it. So we built a really simple design process where you only need to tell us the positions you want to mutate, the substitutions you want to introduce, the liabilities and restriction sites you want to avoid, and we will do the rest. So if you're not exactly sure what you want, our team will work with you to find the best library to meet your needs. And many of our customers are excited by the flexibility of our platform, which enables them to outsource this library build and design process so they can focus on doing their experiments and not the library build itself. Our sold library leverages our technology to synthesize each variant base by base, which enables customers to precisely define the changes that they want and to have control over the codon usage to avoid restriction sites and unwanted motifs so they can focus on screening developable variants. And instead of looking for a needle in the haystack, you now have stacks of needles in front of you. And every library is NGS verified, so you can be confident about the library that you're working with. And most importantly, be able to know, now be able to use that negative data to better understand your target of interest. And many people are now using this to feed into their machine learning algorithms to better understand their target and help focus their next library design. If you want more information, please feel free to go to the Twist Libraries webpage for the product sheet. And if you're interested in getting one of these libraries, the submission form can be downloaded on e-commerce. And if you have any questions at all, please feel free to email us at library@twistbioscience.com. And I want to thank my incredible team and colleagues at Twist for help bring this product to life. I hope this can be a tool that enables you to accelerate your research. So thank you for your time, and let's open the floor to questions.
Lucy, thank you so much for a fantastic presentation. I'm particularly excited about what you just said about the negative data. I think knowing exactly what's in your library is incredibly powerful and something that is where Twist is uniquely positioned to offer to our customers. So this is fantastic. As Lucy said, we're going to now open it up to questions. I see there are quite a few rolling in, so that's good. Let's start off with the first one here. So you mentioned CVLs, SVLs, and now sold, of course. How would I know which library products is best for me?
So if you're not sure which library product best suits your needs, we can work together to figure it out. You can always email library@twistbioscience.com. But for SVLs, generally, if you're interested in just mutating a single position at a time, that would be the best library for you. So for combinatorial libraries and SOLT libraries, you're interested in mutating multiple positions at once. So you're trying to see the effect of these combinatorial mutations. If you're doing antibody discovery, combinatorial variant libraries will be a perfect fit for you. And then with SOLT, I think a lot of our customers are using it for enzyme evolution. But again, it really depends on what you're trying to achieve, and we can, I'm happy to set up a time for us to sit down and, talk and go over, what positions you're interested in, what you're trying to do, and how Twist can help in that process.
Great. Okay, let's see here. What is the maximum size of a stretch or multiple stretches that can be achieved in one library?
So with sold libraries, like really exciting thing about it is that the diversity can be spread out anywhere. And I know with some of the other processes and methods out there, when you're designing these like gene blocks or, primers, the stretch can only be either 30 base pairs long, or flanking those regions, you need to have 30 base pairs or 125 base pairs of non-variant sequence. With the sold libraries, we don't have that limitation at all. The diversity can be localized anywhere, and we've tested builds where we have about 700 mutations isolated and localized in just one region. Now, if you're talking about combinatorial variant libraries and you're asking, how long can that stretch be? With length variation libraries, we've built libraries that have a domain that was around 30 amino acids in length. And if you want something longer, we can definitely find a way to make that possible. And it really is about understanding what you're trying to do. And then, our team will work to find a way to make that happen.
Thank you. You mentioned 2 weeks for these libraries. Is this time frame only for the synthesis without QC, or does this include QC ready to go to the customer?
So, once you submit your design, then we start the ball starts rolling. So for sold libraries, the 2 weeks is as soon as you place the order, and we've ordered the oligos and then we start the build. So yes, around 2 weeks. And of course, if there are issues with it, if there's issues with the build, then that's going to increase the turnaround time a bit more. But in general, what we're seeing is that we're able to do the build in-house in 2 to 3 days, and the rest is just getting it ready for sequencing, getting the oligos designed, and getting them in our hands.
Right. You kind of sort of answered this, but what would be the limit of the number of positions that can be mutated in a sold library?
I would like to say it's a limitless, but as you can see, I don't know if you remember from the last slide, the diversity can be up to 10 to the 8. So I know that there's some people who want a library that captures 10 to the 81 in diversity. Well, that's not possible. So as long as the diversity is 10 to the 8, I think we can find a way to make it work, because the libraries that we've tested in the development of this product, the diversity can be very close to each other or completely scattered. So right now, I would say it's really project dependent.
Right, so we'd encourage you to submit your designs for a quick assessment.
Yeah.
I always like to say, basically send us your dream library and we'll figure out a way to make it.
Yeah, exactly.
What is the typical range of diversity? So I guess this means, what have we seen before in terms of diversity? You already mentioned 10 to the 8, but what is the typical range?
Honestly, we've seen libraries where the diversity is just 1,000, or 1,000 unique variants, all the way up to 10 to the 8. We right now we're not taking anything greater than 10 to the 8 because we've optimized our process for this. But if there's a project coming in where you're interested in just pushing the limits a bit, we can definitely sit down with you and review the project together and see what our team is able to do.
Perfect. Will the multiple stretch libraries cover all possible combinations of the degenerated codons?
So we get that question a lot. When you're using degenerate codons, you do see these codon biases, and I want to reiterate that with our processes, we don't use degenerate bases. We synthesize everything base by base. And so what you can expect to see is a uniform incorporation of all the requested variants in that region. So yes, and there's no biases. And if you want any motifs or restriction sites avoided in that region, we can absolutely do that in the design, because we can pre-screen them and then remove them from your library. So you don't have to worry about any of that.
Yeah, I think this is sometimes sort of a paradigm shift for folks who are accustomed through decades of use of using these legacy approaches. So I think outside the box, we can fit a degenerate design, but we're not restricted to it.
Yeah, and we synthesize each of the variants, which gives us the ability to go through them, on our computers and toss out any ones that contain, motifs that might result in like developability issues. And if one of the variants contain like a restriction site, we can easily swap out one of the codons and use another one in its place so that you can still maintain that diversity and you don't have to discard the entire variant altogether.
Right. Are clone libraries available, i.e. in an expression plasmid? Do these have quality control to ensure diversity in library size?
So right now, this is just a double-stranded linear DNA library. But if you're interested in having these libraries cloned, please talk to me after or send me an email and we can talk more about it. And I mean, that's definitely on our roadmap, and we definitely want to, offer a product that our customer can just take, just receive in the box and just go move right into their experiments.
Right. Here's another question about turnaround time. Is there a limit to residue position and substitutions per position? You sort of answer that. It's design dependent.
Mm-hmm.
We've had folks submit library designs that have 10 to the 81 variants in them. So you could submit whatever you want, and we'll likely be able to find a solution. Is I think what your answer is.
Yeah, it's kind of like what you said, tell us what your dream library is, and our team will work with you to, figure out what we can actually do.
Okay, so there's a few more questions along that similar idea in terms of limitations around design. So I don't think we need to go through each one. Essentially, 10 to the 8 is the maximum diversity spec for these libraries, and how that diversity is spread around is in theory limitless, but is basically design dependent. So we would encourage you to reach out to us at libraries@twistbioscience.com. Another clone question. This is a different take on the clone question. So can libraries be cloned and arrayed?
So if you are talking about a site variant library, yes. For site variant libraries, we offered cloning, and they are arrayed per position. So for a site variant library, if you remember, you're mutating one position at a time. So within that position, you can have anywhere from a single amino acid change to substituting in all 20 amino acids, or one single codon, or all 64 codons. So for an SVL, per position, each well will contain that set of variants. We can't individually array all 64 or all 20 variants individually on the 96-well plate, but we can keep them within its positional set. For CVLs and SOL libraries, because they are billed as a pool, we can't individually array them.
Right. And I've also spoken to people who have this exceptionally low library diversity where, because our gene synthesis is so inexpensive, oftentimes it just makes sense to have each one synthesized one by one. So that's certainly an option depending on the type of diversity that you're looking for your project. Can the library include indels, in-frame or otherwise?
If you are talking about combinatorial variant libraries, yes, you absolutely can do that. And that's the beauty of being able to synthesize each variant base by base. You can tell us exactly what, if you have a list of variants that's been pre-designed and you want us to synthesize all of that and incorporate that into your library, absolutely no problem, we can do that. But with the SOL library, for the applications that our customers wanted it for, we don't do insertions and deletions. But we can definitely talk more about it and see what we can do. It's always exciting to see what customers are interested in working with. And as you can see, with SOL, our customers wanted a library that they can get in 2 weeks and diversity scattered across the wild type sequence, and we made that happen.
Specification somehow at Twist. Okay, that's another. More cloning questions.
So it sounds like cloning is something that a lot of people want,
Yeah.
and I'll definitely take that feedback and bring it back to the team and see what we can do for you there.
Yes, lots of cloning questions, lots of questions on design limitations. So I'm trying to sort through to find one that's not in that kind of one of those two categories. Um, so what is the actual delivered output on ordering a library individual plasmids? Again, no, that would be a pool of double-stranded DNA is the deliverable in this case.
The DNA would be dried down. For soul libraries, you get 1 microgram, and if you need more material, let us know. We can definitely scale it up for you. Same with our CVL libraries. We typically deliver 1 microgram, and if you need more material, like 10, 20 micrograms, 50 micrograms, we've done that before, and we are able to do it in a way that doesn't introduce any bias, because when we know that you want more material, what we do is we use more starting material during the build. So that way we can minimize amplification and bias overall.
Okay, I'm having a hard time finding one that's not related to design limitations. So lots of interest in limitations on how these libraries are designed. So once again, to all you folks who are answering those questions, I don't think we need to go through each one, but please reach out to libraries@twistbioscience.com. We'd be happy to discuss your specific design with you. More deliverable questions. Okay, can the library be barcoded for Illumina sequencing?
We haven't done that before, and I mean, why don't we set up a time to talk about it? I can set up a meeting between you, me, and the team, and we can see what we can do.
So I assume this question is referring to the researcher doing the cloning in their lab, but how do you ensure the library diversity doesn't get compromised due to cloning?
So, so for us, whenever we do, we offer cloning at Twist for the SVLs and the CVLs, and what we typically do is we sequence the libraries before cloning, right? So we want to make sure that all the diversity that's been requested by the customer has been incorporated, and then we move into the next process. So we do the cloning, and then after that we take the clone library and then we sequence it again to check that the diversity is still there, and if we see any changes from cloning, then we'll be able to try to figure out what caused it, and the customer will have information on that. So that's part of what we do here at Twist.
Okay, how can I avoid motifs that are known to cause issues for developability?
There's a few great papers out there that talk about these developability issues and what motifs to avoid. Typically, if you want to avoid those motifs, you have to be really careful when it comes to designing the libraries, right? Because especially if you're using, like trimers and, degenerate oligos, you need to avoid certain combinations of amino acids with each other to avoid those motifs. But, so what ends up happening is that sometimes people just avoid those amino acids altogether, right? Because you can't really distinguish how they're going to assemble together, and how they're going to pair together. But with Twist, what we're able to do is, since we take your design and then we design it all in silico. So we screen them already at the amino acid level for sequences that contain these developability motifs, and then we just toss them out and move forward with just the variants that don't contain any of those motifs at all. So you don't have to worry about these liabilities. And then when you're moving into the next stage of your work in terms of screening, you don't have to worry about how this might take up more resources, because you need to screen through more of these unwanted variants.
Okay, here's a different one. Do sold libraries come with adapter sequences on the ends?
So with all libraries or of all the CVLs and SOL libraries, we add our twist universal tails to them. And, for some people, this doesn't really matter because what they'll do is they'll add on, in addition, so outside of their library, they add on, restrictions flanks that contain these restriction sites. So they plan to digest the library and then clone it into the vector. So with twist libraries, we add on our universal tails outside of those flanks. But if you want to omit that, that's completely possible, and we can just leave that out altogether. So it really depends on what you want, and if you don't want the adapters, no problem.
Great, thank you. There's a couple questions on this general theme as well. So would it be possible to make a bias library?
Right now, for CVLs, absolutely. If you, however you want to play with it, if you want to set one amino acid to 1% and the other one to 23, you can do all of that with our combinatorial variant libraries. With our sold libraries right now, you can't bias it towards a certain amino acid. It's completely uniform. But if this is an area where we are seeing a lot of interest, we can definitely work to develop another version of SOLD where you can manipulate the amino acid distribution at each position. But right now, they're completely uniform.
Right. And I'm actually, after listening to your presentation, incredibly impressed with the level of uniformity that you're able to achieve. I think it's something that's very unique.
Yeah, we have a really wonderful team. They made it possible.
Here's another one. What algorithms do you use that speak to bacterial or mammalian codon bias?
I'm not sure that I really understand the question about what algorithm that we use.
I don't understand the speak to it part, but maybe speak to, can customers use a custom codon table?
Yes, customers can
work with them to codon optimize the sequence and the substitutions that are made.
Yes, so customers, with our platform, because again, we can do everything at the DNA level or at the base level. You can give us any custom codon table that you have and that you've designed or that you've built, and you want us to incorporate that into the library. Absolutely, we can feed that into our design platform.
There's a few questions along the theme of, I'll just try to paraphrase what they say, the size limitation. If they wanted to go maybe a little bit beyond 1 KB, how feasible would that be?
I mean, we can definitely work with you to try it. We've built a few libraries that are 100 base pairs over, and a couple that I think are maybe slightly 200 base pairs over. There's definitely some room for we can be flexible. So again, it really depends on the design. But if you're going from 1 KB to 2 KB, then probably not at this point right now. But if it's just a few hundred base pairs outside of that, I think that should be doable.
Regarding the conversation we're just having on the universal tails, if the tails are present, does it count against the 1 KB?
No, it doesn't.
Great. This is what I love, this question. So this is someone coming to us with their dream library. So I'm interested in building a mutation library on cDNA that is 5 KB in length. What is the best strategy to do that?
Ooh, okay. I think first we need to look at where the diversity is located. And then, since it's 5 KB, I'm not sure if SOLD is the best library for it, but we can probably do something either with, SVLs or CVLs, but definitely need to take a look at the sequence and the design itself. I mean, we can find ways to piece them all together, but until I have an idea of what the design looks like, I can't make a confident, statement about what approach we can take.
Great. Just tons of questions, lots of interest on how these things can be designed. Still coming in. So at this point, I would say, again, I would encourage you all to reach out to Lucy or libraries at twistbioscience.com to get any specific questions about your design limitations, if there are any. But generally speaking, outside of the aforementioned 10 to the 81 library, we're willing to take a shot at anything you've got, and we probably have a technology solution meet your needs. Just one more. So here's another question that's kind of interesting. So can you get a small library, for example, 384 variants, as separate variants rather than a mix? So again, I think that this would be, we would work with the gene synthesis group to probably answer this question.
Mm-hmm. Yeah. SOLD and CVL are all delivered as pools because they're built as pools, but with site variant libraries, they're the only ones that can be delivered as individual variant sets.
Right. Okay. So everyone else, again, who asked questions about design limitations, bias, how much constant sequence needs to be present between positions, how many consecutive variable positions can you have in a single stretch? The answer, in short, to all of these is there's really no limit. The strength of the platform is that we have unprecedented access to extremely high quality oligos, and we can mix those together to generate a library that will likely meet your specifications and deliver to you all of the variants that you're looking for. So at this time, I would like to remind everybody that once we close the webinar, you're going to be directed to a brief survey. Looks like we're giving you about 8 minutes back, so we're extremely grateful if you would take that time to answer the survey, help us improve future webinars like this. Lucy, thank you so much for your hard work to lead the development of this product at Twist and their presentation today. I, for one, am extremely excited that we have added to the already impressive lineup of library products at Twist Bioscience. So good job. Thank you so much. Thanks everyone for coming and have a great day.
Thank you.